Good evening, everyone. First of all, thank you to the organizers for inviting me for the presentation. So today I will be talking about one of our recent research articles. And first of all, I'm a doctoral researcher at Institute of Biomedicine in University of Turku in Finland. Today I'll be talking about fibroblast growth factor receptor like 1 protein in prostate cancer progression, its role and its behavior. So first of all, coming to the basics on fibroblast growth factor receptor signaling pathway. So first of all, there are 5 FGFRs. So FGFR 1 to 4 and FGFR L1, which is discovered around 2000, and not much is known about it. So in this FGFR 1 to 4, it has an intracellular tyrosine kinase domain that phosphorylates FRS2 alpha, which in turn activates the PI3-AKT, MAPK, hedgehog signaling, which help in proliferation of the cells and cancer progression. And FGFR L1 doesn't have this intracellular tyrosine kinase domain, but it is shown in many studies to be involved in this FGFR pathway. So coming to the comparison of these structures. So what is the difference between this FGFR L1 and the other 4 FGFRs? So first of all, like I said, FGFR L1 doesn't have this kinase domain, but the extracellular domain it has similar structure to the other FGFRs. Thereby, the FGFs, those are the ligands, they still bind to FGFR L1. Now coming to why did we pick FGFR L1 in our studies? So we collected around 150 patient samples from Turku University Hospital in the form of tissues, so prostate cancer tissue samples, and we checked the mRNA expression of this FGFR L1 in them, and we saw that in cancer samples, the mRNA of L1 was higher as compared to the adjacent benign sample of the prostate. This got us interested to know more about why was the expression higher in cancer? So like any other studies, so the first thing we do is try to find model systems that could replicate the cancer in the in vitro environment in laboratories. So we had, there are various prostate cancer cell lines that are available, like we have PC3Ms, we have LNCAP, VCAP, 22R1s, etc. So what we did is we tried to knock down FGFR L1 expression in these PC3M cells, and we successfully knocked it down, and we created 2 stable knockdown clones. So we have the knockdown clones of L1, and then we have the control clones, which have the expression of L1. And then we use these in both 2D and 3D studies. So in 2D cell culture studies, we observed that the cells where there is knockdown of L1, they grew slightly faster as compared to the control cells, which have L1. Similarly, the migration rate of the cells of knockdown was slightly faster as compared to the controls. So this was in 2D cultures. Now, the similar results were seen in 3D cultures, where we used these cells to grow them as organoids in matrigel. And we could see that here in the picture, FGFR L1 spheroids were slightly bigger as compared to the controls. And this was quantitated using the AMIDA software that is made in the lab. And we could see that the area of these L1 knockdown organoids was bigger. So that was regarding the functional studies. That's the confluence of the cells and how fast they grew. Now coming to the signaling, what is happening when L1 is lost in the cells? So as shown in the first slide, FRS2 alpha is one of the first protein that gets phosphorylated in FGFR pathway, and that's the important protein because after that is when the downstream pathways are activated. So what we did is in the same cells, we overexpressed FGFR L1, and on overexpressing this protein, we see that the phosphorylation of this FRS2 alpha is inhibited, whereas when we knock down FGFR L1, the phosphorylation of FRS2 alpha is increased, showing that FGFR L1 could act as a decoy receptor and prevent the function of the FGFR pathway. So that was the in vivo studies in the lab. Now we want to see what happens I mean, that was in vitro studies, I'm sorry. Now we want to see what happens in vivo. So we made xenograft tumors using these cells in mice, and we saw the opposite effect. So that is when L1 was expression was down, the cells grew faster in vitro, but in vivo, when L1 expression was down, the cells didn't grow at all or very less. So the tumor size was less. So was the body weight. Now, why was this discrepancy in the results in vitro and in vivo? To answer that, we went ahead and did RNA sequencing of the in vitro cells and the in vivo tumor cells. And when we did RNA sequencing, there were many genes that were upregulated, downregulated, and here they are. So the genes that are downregulated in xenografts, those are upregulated in xenografts. The ones that are upregulated in both cell lines and xenografts, and the ones that are downregulated in both cell line and xenografts. So from these, we got some hints, because here we see the genes that are in boxes, SMAW, SERPIN, CXL1. These are the genes that are involved in tumor stroma interactions. And these help in proliferation of the cells. So, and these were downregulated. So indicating that this could be one of the reasons why the tumors in vivo did not grow. Similarly, genes like BMP7, NCAM, TIMP3, these are also involved in tumor stroma interactions, and these genes were upregulated, but these are tumor suppressors, and these were upregulated. So therefore, the in vivo xenografts did not grow again. And particularly a gene PTX3. Now, pentraxin 3 is a gene which is a FGF2 antagonist. So it doesn't allow the angiogenesis that FGF2 plays a role in, again showing that it's upregulated, because of which the xenograft tumors did not grow. So we found explanations here. And of course, the tumor stroma interaction is much better in a xenograft model than in 2D or in vitro cultures. Now, that was the functional studies and what happened on L1 knockdown or overexpression. Now, coming to the localization. So basically, FGFR-L1 is a membrane protein. It is present in the membrane of the cells. So here we see that FGFR-L1 in vitro, it's present in the membrane of the cells. But when you knock it down, there are some expressions remaining in cytoplasm, but nothing in the membrane. Similarly, in xenografts, we can see that L1 is present in the membrane in control cells, and in knock it down, there is no expression of it in the membrane. Now, how does that the localization of L1 seen in the patient samples? So coming back to the first clinical samples that we had of the patient tissues, we saw that the membrane expression of FGFR-L1, it decreased as the cancer progressed. So from prostate cancer, it went into the advanced stage of prostate cancer, and then even more advanced stage, that is castration-resistant prostate cancer. Whereas the cytoplasmic expression of L1, it increased. And the nuclear expression of L1 was also slightly higher and found in the most advanced stage of prostate cancer. So therefore, the localization of L1 was changing as the cancer was progressing. And what was the impact of this localization? So what exactly happened when the membrane protein started entering the cytoplasm and then the nucleus? So for that, the Kaplan-Meier plots were seen, wherein we see the biochemical recurrence of prostate-specific antigen. So if this antigen recurs in the blood sample of the patient, that means the cancer has relapsed. And we see that based on the membrane expression of this FGFR-L1, there is not a significant difference in the survival of the patient. Based on the cytoplasmic expression of L1, again, there is not a significant difference in the survival of patient. But based on the nuclear expression of FGFR-L1, there was a significant difference in survival of patient. And the more L1 in the nucleus, the lesser the survival of the patient, indicating that this could act as a prognostic biomarker. So to conclude, we have used different model systems, the patient tumor tissues, the in vitro organoids and 2D cultures and xenografts to study the L1 action. And we see that L1 is overexpressed in prostate cancer, Particularly, it starts entering into the cytoplasm, from the membrane into the cytoplasm, into the nucleus. The cancer progresses, and once it's in the nucleus, the survival of the patient decreases. So I would like to acknowledge my supervisors, Professor Matthias Snees, Dr. Malin Akkerfeld, and Professor Pirkko Harkonen for guiding me through this study, and the other group members and co-authors which were involved in the study, the funding agencies which have provided us the funding to complete this project. Thank you. I'm open to take any questions if there are any.